let me now switch gears and give you the kind of a bigger vision what is happening throughout the field, not only what is happening inside the room looking into the quantitative proteomics. Okay. So, that is just the one part of the bigger picture what is we are studying here, but if you really want to obtain meaningful information, you have to be much more broad, and therefore, as I mentioned, my lab uses a variety of platforms, not only the mass spec based platform, but even within proteomics there are a variety of platforms which one need to be very open to use. And depending on what question you want to ask, you can utilize the right platforms. Right now I am just mentioning to you like the systems network information, which one could obtain from variety of ways of networks, modelings, computational based information, and these systems network can only come if you have the access to the large omic kind of data set at the variety of levels, especially if you think about your different approaches which I talked in the very first lecture as well, that if you have information from the genome to transcriptome to proteome, metabolome and phenome, then probably you could build that information at the systems level, and to glean into the actual biological problems, one need to do a lot of computational analysis, modeling analysis, building those network, and then you can find the right answer to the problems. So, what I will do now, I will kind of just give you some very intriguing thoughts about what is happening elsewhere in the world and the kind of revolutions in progress. And I think we are very fortunate, and we are in an area or in an era where we live, where all kind of new things are still in the process. Okay, it is not that you have already learned that you can do a cloning, and you can do how to learn the cloning from a Sandbrook book, right? So, it is not that you have to really still develop new assays, you have to still develop new technologies to find the answer. So, you are at a part when you are the part of the revolution in progress, and there are a couple of things which I thought to mention to you here. One is precision medicine. Okay, precision medicine is how accurate your targets can be to define the right disease state, and therefore, in US they have very recently launched a big initiative which Barack Obama kind of made announcement. I will just show you this highlight here.
This is going to be completely unprecedented. It is going to affect medicine in ways we've never seen before.
You can match a blood transfusion to a blood type. What if matching a cancer cure to our genetic code was just as easy, just as standard? And that's the promise of precision medicine, delivering the right treatments at the right time, every time, to the right person.
Precision medicine enables the ultimate delivery of personalized medicine, which means that you come to your doctor, you have your genetic information, and the doctor can use that genetic information to specifically design treatments for you. But obviously, there will be thousands of individuals that have your disease and your gene, and so it's personal for you, but it's also treating large numbers of people in the same situation. We can look at individual tumors. We can even look at individual cells within tumors and understand how their system has gone awry. It's possible to analyze this very complex data using state-of-the-art computer algorithms to be able to see the interaction of proteins in a cancer cell and to design drugs that will inhibit or treat them. A really good example of how all this works is with a drug called Gleevec, and this is a drug for a certain type of leukemia. This drug had a miraculous effect. I mean, it basically cured the disease. However, the individuals who received this 6 months, a year later, it was back. Then a second drug was developed. We had the tools to go back, look at the sequence of that reverted protein and know where the change was, and then design a second drug to address that. And now the combination of those two drugs that are used in sequence basically has cured one form of this disease. And so I think that's what's going to happen. There'll be more and more examples of cases where we can join genetics, biology, and medicine.
All right, so not only proteomics is the ultimate, but of course there are various complementary technologies which has made significant progress, and genomics of course has been the leading in that way, being all the genome sequenced in around 2002 or so, and then availability of next generation sequencing platforms now, which has made it very robust platform now, and the cost has also got drastically reduced to sequence each genome in less than $1,000. So based on that, precision medicine currently is more aiming towards looking at the genetic mutation and individual variations at the genome level. And US government, Barack Obama recently announced this particular precision medicine area. Whereas of course looking into the success of the precision medicine using genomics, precision proteomics is the next way when proteomics field is progressing, and of course a lot of things which you are learning in targeted proteomics is catering towards that goal. So this is what precision medicine initiative Barack Obama mentioned, that because each individual, each patient is unique, doctors are always try to tailor their treatments as best as they can do as an individual. You can match a blood transfusion to a blood type. That was very important discovery. But what if matching a cancer cure to our genetic code was just as easy, just as a standard? What if figuring out the right dose of medicine was as simple as taking our temperature? So far these diseases have been very complex. We cannot measure them as easily the way you do any blood test or you do your temperature measurements. But if the individual patient therapies in precision medicine be possible, which looks like it's going to be reality now, things will become very different. Another initiative which is happening is P4 medicine, looking at the systems level information again. The way the field is progressing towards the predictive, preventive, personalized and participatory, and the major lead for this part of project is coming from Institute for System Biology, ISB at Washington, and Dr. Leory Hood, he is the pioneer in that area. Let's listen from him, the emergence of P4.
Over the past 12 years, there's been a profound revolution in biology. That is, we've started making systems approaches to the complexity of biology, approaches that are holistic and global in their nature. And the application of this systems thinking to medicine, which started perhaps 10 years ago or so, has created both what I call systems medicine and the application of systems medicine to patients, something that I call the P4 medicine revolution. And a really important point that I'd like to make is the P4 revolution, personalized medicine really on steroids, comes not only from the convergence of patient-driven social networks and the convergence of big data, a lot of which you've heard about here, but it comes from the convergence of systems medicine. I think it's really important to point out that in many of the national publications, The Creative Destruction of Medicine by Eric Topol, he beautifully outlined the network and the big data aspects of the new medicine. He didn't talk about systems medicine. So what I'd like to do is give you a real sense of what systems medicine is all about and how its translation to patients in terms of P4 medicine is really going to be revolutionary. My view in the world is that each individual patient in 10 years will be surrounded by a virtual data cloud of billions of data points of enormously heterogeneous types, ranging from molecular all the way up to social networks. And the reason we need this enormous amount of data is the incredible complexity of biology, and by inference, disease, a complexity that arises from the normal chaotic process of Darwinian evolution. The points I would make about these big data are, number one, it is going to be absolutely critical that we can integrate these data together in means that are appropriate to create predictive models that can sculpt health and wellness for each individual. And number two, we have to realize that the challenge for all big data is the signal-to-noise problem. And I'm going to emphasize the noise problem can't be solved alone by machine learning. Rather, you have to integrate with machine learning enormous domain expertise. And I'll show you an example of that. So systems medicine deals with this complexity by thinking about biology as an informational science, by creating an appropriate infrastructure for generating effectively large amounts of data, by creating the experimental approaches that can be holistic in nature, and then having each of those drive the emerging technologies that open up new areas of data space and their corresponding analytic tools. So let me make 3 points about medicine as an informational science. Number one, there are fundamentally 2 types of data in living organisms: the digital genome and the environmental signals that come from the outside. Together, they blend this information to create the phenotype of the organism. And of course, what connects the types of information with phenotype are 2 informational structures: biological networks that capture and transmit, integrate, and finally pass off to the molecular machines that execute the functions of life, the information that creates phenotype. And what is the central focus of systems approaches are the dynamics of these 2 types of informational structure. And the other point that I would make is that biological information is obviously multiscalar: DNA to RNA to proteins to networks to cells, all the way up to organs and individuals, populations, and ecologies. And the critical thing is at each of those levels, the environment imposes new signals on how the information is used, and accordingly, what is necessary is to capture as many of these levels of information as possible and integrate them in a manner that explicates the nature of the environmental signals. For to completely understand the complexity of systems, you need to understand the digital input and as well the environmental input. How you do these integrations is one of the grand challenges of systems biology. I would argue one of the most powerful aspects of systems approach are the appropriate infrastructures that are created. When we started the Institute for Systems Biology 12 years ago, the mantra we had was leading edge biology mandates the need to develop new technologies. They in turn mandate the necessity for new analytic tools. And that the biology seamlessly drives technology and computation in ways that can revolutionize the biology. To make that work, you have to have a completely cross-disciplinary environment with.
So what is the most important and interesting fact from his talk and of course his work is that not only one need to measure the the problems happening from the healthy versus diseased, but what is happening in the same healthy individual as well. Looking at your wellness profile. So the new project which they have initiated is on the same individual, look at entire omics profiling, look into the genome to the transcriptome, proteome and metabolome, integrate those information of the same individual at different time points, and then you can try to predict, is there everything happening okay in the body, are there some disease could be indicative, some pathways might be changing, indicative of a disease might be prone, and that is a wellness study. So in which way P4 medicine can transform, and the medicine is actually is really being led by this group and their center, and now there are some certain companies which has come forward to take the P4 medicine for the wellness studies. It is personalized approach, yes. But how to do the systems network and generate that information coming out from the omics data was the key from this work. Another major initiative happening at the looking at the metabolites of individual, and this project is happening at the Imperial College London on the Human Phenome Project. In fact, the leaders of this project were recently in IIT Bombay for this our workshop. Now you have talked, we talked about precision medicine on the genome level. I told you how to integrate those using the P4 medicine. This initiative is happening only at the metabolomic level. Large number of metabolites being measured on hundreds of thousands of individuals using both mass spectrometry and NMR kind of platforms to look into their serum, their urine, saliva, all type of body fluids and their interaction with the environment. So that is the Human Phenome Project, which was one of the mega funded projects from UK government, along with some support from London Olympics, what they obtained, and the 10 million pound grants. Now this is a real at high throughput level this project happening with several mass spec and several NMRs to measure various metabolite changes happening in the individuals. And Dr. Jeremy Nicholson and his group from Human Phenome Project have recently published one paper in the Science Translational Medicine, where they have shown the entire networks being modulated in individuals when they measured a large number of patients for the urine samples. Again, these kind of information is being generated now, linked with the clinical information, linked with the environment factors is getting very important to provide the right cues.
Key objectives in 21st century medicine are to understand and improve personalised healthcare and to understand and improve the population health as a whole. And individuals and populations, their biology and also the likelihood of them getting a disease is affected by the gene to environment interaction. And those gene-environment interactions not only change the individual probabilities of getting disease, but also they change your metabolism. So the metabolic phenotype effectively is where genes and environment meet. So it gives you a readout on somebody's biology that isn't just captured by the genome alone. So what we're trying to do here at Imperial, and have been for a number of years, is apply a whole range of advanced technologies, things like nuclear magnetic resonance spectroscopy and mass spectrometry, for metabolically profiling complex samples such as urine and plasma and tissues, biopsies and that sort of thing, in order to get profiles that relate not only to disease processes but also to disease risk factors. And we've established a very large centre here to do exactly that. Recently, because of the Olympic Games, there's been a drug testing facility that's been created to screen athletes for illegal substances in relation to their sporting pursuits. The analytical technology and instrumentation is very similar to that's required for phenotyping on a large scale. So we've been successful in putting together a proposal with King's College London for taking over the drug testing facility at the end of the Olympic Games and to make this equipment and instrumentation available to the UK university population for doing large-scale metabolic phenotyping. So the technologies that we've developed here at Imperial, we will adapt and develop for this much wider use by the community.
The need it's addressing is that all over the country, many clinicians, scientists, epidemiologists have been collecting cohorts of patients with particular conditions or large-scale population cohorts with a very large number of banked samples. And the genetic determinants of those have been addressed by GWAS and other large-scale studies, and have given some often very interesting hints, but also shown that a great deal of disease remains unexplained at the moment by genetic basis, complex trait, and so forth. Where this new facility comes in now is to look very much at the effects of genes and the environment acting together by looking at metabolic profiling, and offers us the ability to do this at scale and hopefully at very low cost for individual sample study. And we think that this will address a very large number of researchers across the UK and indeed more widely across Europe and North America, who we think will wish to take the opportunity to use this resource together with the bioinformatics behind it and the scientific expertise of Jeremy Nicholson's group.
There are a number of different outcomes in scientific and medical terms that come from this type of phenotyping activity. The first one is to improve our knowledge about population health in general by metabolically classifying individuals from large population studies and linking those into potential disease risks of the future. And if we understand more about those gene environment interactions that create health problems, then we can legislate better and advise people better about their lifestyle management. So that's a major outcome of this sort of activity. We're also going to be discovering new biomarkers that relate to disease processes, and those biomarkers relate to mechanistic processes in cells and in the body, which will give us insight into the disease itself and then potentially get new methods of attacking those diseases, new therapeutic targets. There's another angle on this is to do with technology. So we have important strategic partners, Waters Corporation in mass spectrometry, in Bruker spectrospin, in nuclear magnetic resonance, and these partners are putting in a lot of investment into developing the next generation of analytical technology. And so we're talking about miniaturization, increased use of green solvents, because when you do large-scale analyses of these sorts, you use a lot of chemicals, and we want to make that more financially efficient and also better for the environment. And also all the technologies that we're developing here and the computational methods for extracting information are highly relevant to other areas of analytical science relating to environmental analytical research, pesticides, pollutants, food science. The list is really quite comprehensive. So in addition to addressing a series of medical needs and problems solving to do with population health, there's going to be a long-term legacy of this work in terms of analytical science for the United Kingdom.
Ideally, this study has started showing its kind of bigger impact because initially it takes a lot of investment to do this kind of large profiling. It takes very dedicated huge servers to save this data. Just imagine the same samples being analyzed from NMR and from mass specs, and then how much several TB data you are generating from each experiment. Storing those, having the right databases and linking those with the clinical information. With those, I think they have really started reaching out to the right information now, and several series of very good papers that are coming out from this group and this work. And their intention is to also spread out these kind of human phenome centers in different parts of the world, just so that this kind of technology could be utilized for doing metabolic profiling. Targeted proteomics is another revolution in field in progress happening. It was known as the nature methods of year 2012. So technology is just kind of emerging, only 3 years old. And in this area, which you have seen that the major question of proteomics critics have been that why proteomics, although it is giving lot of data, but it is not impacting the clinical research, translational research. What is the reason for that? So from one of the papers, Dr. L. Edwards, what he makes conclusion, which is kind of very good summary of why proteomics has not made huge impact in the field, is we have always been trying to validate the proteins for which we have antibodies available. And antibodies companies have raised for those targets which are highly abundant, right, and for very few targets. So whenever you need to validate your proteins, you are always linked to the antibodies available commercially or in the market. And so always you are going to look for haptoglobin, you are looking for hemopexin, you are going to look for those proteins which are for which you have good antibodies available. Whereas in your mass spec data, you will have many proteins which are kind of very unknown, which are showing you huge fold changes, but you have no way to do validation for those. And therefore those proteins die because you are not able to validate. Until you validate, nobody is going to rely on your data. Therefore, in absence of targeted proteomics field, which actually aims to look into the mass spec data further and do validation from the peptide level information. So far the field has been relying on the antibody based assays. You talked about immunohistochemistry, you talk about ELISA, you talk about Western blots. All of these are depending on antibodies, good antibodies. So the field has not made major impact because there was no good validation strategies available. And what it rightly mentioned here, new findings are reported predominantly on those proteins that are routinely measured in the many labs for which good antibodies are available. So that has been the major limitation why proteomics has not made impact as strongly to clinics as it should have. Now with this targeted proteomics approach, you can have the set of predefined peptides which are very abundant in the given disease, for which you consider them as a potential biomarker. Now you can take those and generate very specific mass spec based assays directly for those. So you are doing discovery and quantitation in mass spec, and you are also doing validation using mass spec. There is no role of antibodies in between, and therefore what you discover, you are validating that. Otherwise, you are discovering something else, you need to generate some new reagent for those, then you do validation independently, and many times it fails into that pipeline. So many of the good hits and good targets which people had, now there is another opportunity for them to start validating, and in fact it has started making good impact, and couple of markets are now being approved from US FDA and other regulatory agencies. So therefore there is a good hope that this field is going to make impact.
Improving human health by unlocking the mysteries of diseases is a goal that many scientists share, and it will be enabled by mapping the proteins in the human proteome. Advancements in proteomics analysis are offering people new hope for the development of more effective drugs and the promise of protein biomarkers for a more personalized approach to medicine. At the Swiss Federal Institute of Technology, known as ETH Zurich, in collaboration with the Institute for Systems Biology, scientists have achieved one of the most significant advancements in the proteomics field to date, generating mass spectrometry assays for each of the proteins that correspond to the 20,300 genes in the human genome.
With the MRM Atlas project, we try to basically provide a resource to the scientific community so that we develop these assays once and for all, and then people can download these assays. The ultimate goal is to apply proteomics to biological and clinical research to try to understand how biological processes function and are controlled by measuring the proteome.
Importantly, the MRM Atlas expands into never-before-seen areas of the proteome. Scientists will now be able to analyze dramatically more proteins in the human body with greater specificity, speed, and reproducibility than ever previously possible. The completion of the MRM Atlas marks an important turning point in the field that is expected to affect biomedical research for years to come.
We have been now for a few years working very hard on developing and introducing targeted mass spectrometry. It's gaining, I think, a lot of attention and traction. It's getting fairly widely used. The technique that supports it is referred to as selected reaction monitoring, also frequently referred to as multiple reaction monitoring. In contrast to immunoassays, we don't need to inject any animals, but we need to generate spectra in a mass spectrometer such as the Q-trap mass spectrometer. The Q-trap technology is an essential part for us to support targeted mass spectrometry by SRM. And the Q-trap instrument is the only instrument that can generate full fragment ion spectra in a very high throughput mode and high sensitivity, and as well as then use these spectra or the transitions derived from it to do SRM measurements. So in that sense, it's an ideal instrument for us because we spend a lot of time developing large-scale assays and then to use them for the measurement. We can do this all on the same platform. AB SciEx has consistently produced high-quality, innovative instrumentation, also chemical reagents that are useful in the context of proteomics, which is expanding and will be driven by new questions that can be asked.
And AB SciEx will be there to deliver the answers.
All right, so Rudy Ebersold is one of the pioneers in the field at ETH Zurich, and his lab was kind of instrumental in developing this SRM and MRM assays. And so as the ISB, Institute of Systems Biology, Dr. Robert Morris and his group, they developed SRM Atlas. Brendan McLean from Institute of Washington, they developed Skyline. And with all of these developments happening parallel, now this is looks like a new way this field is progressing now, where biomarkers can be validated at the protein level. And in the United States, National Cancer Institute and CI, they have now launched this new project from CPTAC, which is Clinical Proteomic Tumor Analysis Consortium, CPTAC. They have now launched this in a precision proteomics project, where these kind of SRM assays being developed for various proteins. So these transition information for various peptides, you can obtain, download from their server, and then you can utilize on your own samples and you can do the validation. So there is a dedicated team of the scientists and the software analysts who are already developing these assays now. The way you used to get the kits for ELISA, now you can get the information for the SRM assays for the given proteins, and you still need to adjust the collision energy and you need to adjust the conditions to do validation. But now things will be much more robust because there is a dedicated project in place where you can obtain lot of clinical assay information from the SRM and MRM assays. Well, so in conclusion, in general, lot of changes at the genome, transcriptome, proteomics is giving rise to system biology information, which is going to be informative and linked for the physiology and medicine. while we have been looking at a proteome level information which has been very daunting, very challenging, being very dynamic and very low abundant proteins relevant for diseases, proteomics in general has evolved quickly to make an impact not only in the physiology and medicine, but in variety of other application in all over the life sciences and biology in general. However, until unless we integrate this protein information with other omics information, I do not think we can really unravel any of these complexity at the real biology level. So, we need to be very open to integrate information from other omic level as well. So, that is kind of conclusion from this presentation.